Classification of amino acids based on R group. By the end of this lesson, students should be able to classify amino acids based on the chemical properties of their side chains, explain the psychochemical properties of each group, nonpolar, polar uncharged, acidic, and basic, discuss how R groups influence protein structure and function. Lastly, identify amino acids by their structure and assign them to the correct classification.
Amino acids and proteins. What is that?
Proteins are the most abundant macromolecules found within living cells. The protein molecule is composed of hundreds or even thousands of amino acids linked by peptide linkage.
But how do we classify which amino acids they are?
Good question. But first, we have to identify the different parts of an amino acid. All amino acids has an alpha amino group, an alpha carboxylate group, an alpha carbon atom, and the most important of them all, the side chain R group. The side chain R group is what we will use to differentiate one amino acid from the other. Amazing, right? Would you like to know more? Yeah! Great! There are 20 different amino acids. Let's get to know them all. Starting with nonpolar aliphatic amino acids. But before that, you must remember that in order to differentiate each amino acid, you must look at its side chain R group.
But sir, where can we see it?
Just look at the side chain below the alpha carbon, like this one example right here. This amino acid is called glycine. Okay, back to nonpolar aliphatic amino acids. These amino acids have hydrophobic side chains that avoid water and stabilize protein structures by aligning towards the interior of the protein molecule.
What amino acid belongs to that group?
I'm glad you asked. It's glycine. This is also the simplest amino acid.
Is that it?
Glycine only has a hydrogen atom as its side chain, providing flexibility to protein structure. It can be found on the inside or outside of protein.
Does nonpolar aliphatic amino acid only contain glycine?
No, it also has alanine, valine, leucine, and isoleucine. These amino acids have alkyl side chains of varying lengths, contributing to the hydrophobic core or proteins. It also has methionine. This amino acid contains a sulfur atom within a thioether group, and it's often involved in initiating protein synthesis. P is for proline. This amino acid is unique due to its cyclic structure that incorporates the amino nitrogen, restricting flexibility. It's often found in turns and loops of proteins. Question!
Aside from nonpolar aliphatic amino acids, what are the name of the other groups?
Nonpolar aromatic amino acids.
These amino acids have aromatic rings in their side chains, which can absorb UV light, making them useful in determining protein concentration via spectroscopy. Examples include phenylalanine. This amino acid is nonpolar, with a benzyl side chain contributing to the hydrophobic core. Tryptophan. This is the largest amino acid, with a complex double ring that includes a nitrogen atom and plays an important role in protein-protein interactions.
Does that mean we also have polar?
Polar aromatic amino acids.
Tyrosine. This amino acid is polar due to a hydroxyl group attached to the benzyl ring. Its structure is vital to signaling pathways using tyrosine kinase receptors.
We've talked about two groups already, but after we have three more. What are they?
That's right. Let's tackle the other three groups one by one. Polar neutral amino acids. These amino acids have side chains that can form hydrogen bonds with water, making them hydrophilic. Examples include serine and threonine. These both contain hydroxyl groups which are often involved in phosphorylation. Cysteine. This amino acid has a thiol group, making it reactive and prone to forming disulfide bonds, which are crucial for protein stability. And asparagine and glutamine. These contain amide groups that do not gain or lose protons, remaining neutral in charge and often involved in hydrogen bonding.
Neutral? So does it mean we also have charged amino acid?
Yes, these amino acids can be further divided into acidic and basic amino acids based on their side chain charges. They are often important for enzymatic activity and for creating electrostatic interactions that help to stabilize protein structure. Let's talk about the acidic amino acids first. Okay, acidic amino acids are also known as negatively charged amino acids. We have aspartic acid and glutamic acid. These amino acids have carboxyl groups in their side chains, which lose protons and become negatively charged. They are often involved in active sites of enzymes.
Sir, how about the basic amino acid?
Well, basic amino acids are also known as positively charged amino acids. First, we have lysine. This amino acid has a terminal primary amino group, often involved in binding to negatively charged molecules. Next, arginine. This amino acid contains a guanidinone group, making it highly basic and often involved in protein binding. Lastly, histidine. This amino acid contains an imidazole ring, which can shuttle protons, playing a crucial role in enzyme active sites and buffering. We're done classifying amino acids based on their R groups. Did you learn something?
Yes, sir!
To prove that you really learned something, try classifying this amino acid. I'll wait. The answer is... If you got it right, don't forget to comment it down below. A prize is waiting for you. Thank you for watching.